So my name's Moin Saleem. I'm a kidney doctor. I'm a paediatric nephrologist. Work down the road at the Children's Hospital, and I also work as a director of Bristol Renal, which is a big renal disease research group. And what I want to show to you is how we've travelled this journey from an academic, a clinician, to spinning out a gene therapy company, to really trying taking what we've been working on for 20 years into patients, into a translational and hopefully a sea change in the treatment of patients with kidney disease. So some background. Kidney disease is a huge unmet medical need, and I won't sort of go through all the figures on the slide, but it's said that roughly 1 in 7 or 1 in 8 of the world's population has chronic kidney disease, and the costs are huge, and it's an increasing cost, and actually the consequence of chronic kidney disease is dialysis and transplantation, which is available only to a very small section of the population worldwide, so the costs are a severe underestimate of what could be needed. So it's a problem. And I'm going to unashamedly or unabashedly give you a little bit of a background of kidney disease and what the kidneys do. So the kidneys, I usually call them the world's most sophisticated filter, but it does much more than filtering. It's a sort of physiological homeostasis that the kidney achieves in lots of different areas of physiology. So basically the kidney consists of nephrons. We each have about 1 million nephrons, and at the head of the nephron is this unit called the glomerulus, which is a specialized capillary bed. And the glomerulus is really the kind of heart of where most kidney disease originates. And it's 90% of end-stage renal disease, so chronic kidney disease leads to end-stage renal disease, originates in the glomerulus. And the central cell type, the glomerulus only has 3 cell types, and the central cell type of the glomerulus is called the podocyte. And so that opens a door to a specific target that could target gene disease via that crucial cell in the glomerulus called the podocyte. And what you can start by looking at is single gene disorders, so diseases that are caused by inherited mutations in genes, but also then expand that to things like immune disorders, so use the podocyte to regulate local immune activation, and many types of kidney disease are caused by inflammatory glomerular disease. And then there are also a huge group of disorders that are metabolic, so diabetic nephropathy is the commonest cause of kidney disease worldwide, of kidney failure worldwide. And the podocyte is a uniquely insulin-sensitive cell that is also central to the disease of diabetic nephropathy. So why Bristol Renal? What's our unique selling point, if you like, our USP? So we're pioneers and leaders in the field of podocyte biology and have been for the last 20 plus years. We were the first, for example, to develop the human podocyte cell lines that are now standard across academia and industry, and we've licensed them out through RED to industry and academics worldwide. And we're a very large renal research group. We have a reputation for our innovation, and we continue to innovate in the models that we create of human diseases. And we are also sort of setting up and leading not just lab work, but cohorts of patients, international and national cohorts of patients, particularly with rare diseases and also the more common diseases, because in order to be able to study disease, it's not good just to have laboratory models. The laboratory models have got to be accurate, but actually to be able to use patients as part of your research studies, and sometimes I say as the best models of disease, you need to have access to them through really good, well-run cohorts. So where do we take that? So we've got really nice data from an academic lab. What do we do with it? So this is effectively how it happens. So we've got a fantastic university commercialization director who I know well through other sort of industrial liaisons that he's helped us with. So Andrew Wilson, who's here in the audience. And Andrew saw the potential of this immediately. And what Andrew did was get some money from RED, get some consultancy from the Cell and Gene Therapy Catapult in London. They prepared a briefing document. They helped us attend an advice meeting with the regulatory authorities, and that resulted in a non-clinical plan, so a roadmap, so to speak, to take us from that mouse model to a first-in-man study that would be potentially approvable by a regulatory authority. So that gives us the credibility that this could work. So from there, we then needed to have the money to make it work. And we have various industry collaborations around the world, so we're well connected with pharma. So one of the options was pharma and rare disease companies. And the sort of pros and cons of approaching these people and what they offer. So pharma are usually well funded, but it's a different business model from these kind of rare diseases or gene therapy. And they had, particularly most of the ones we talked to, had a relative lack of experience in things like design of the clinical development pathways and manufacturing, which is absolutely key, which I'll come to a bit later. Venture capitalists, slightly different model, if you like. They have quite deep pockets, different models of working with academics, which was important to me because I wanted to stay very much involved. And I found a very distinct approach between the US and the UK companies, which I thought was interesting, and varied levels of experience with gene therapy companies. In the end, we agreed a deal with a venture capital company called Syncona, which some of you may have heard of. It's a UK company spun out originally from Wellcome Trust. And crucially, they already have 5 successful gene therapy companies in their portfolio, so they really had the experience and the know-how. And so they've got a track record and a small and focused team that really likes working with academics, and that was important to me and I think important to the company. And also the foresight to fund this all the way through to clinical development, so not step by step, but they would fund 2 or 3 programs all the way through to first in man, and that takes a lot of the pressure off in the way that company builds. And they're very ambitious. They want to apply the technology to a range of kidney disorders, not just the rare indications, but to include commoner diseases. So they set us up as Purespring Therapeutics. So vision, as I say, introduced clinical therapeutics for the first time via the podocyte with this AAV technology. And if it's successful, this could be really game-changing. So the potential for curative therapy for a number of different kidney diseases is unlocked. So as I say, that's the world's first, as Andrew said, it's the world's first kidney gene therapy company. And the Series A investment, as it's called, was 45 million. So it was their biggest ever Series A investment. And some kind of reflection from my academic perspective of what this takes. So what was the important facets of an academic partner of our group that makes this partnership feasible? And I think very important things are the track record, so to show that you've got leadership and world leadership. These companies are very, very critical and apply a very high critique in the level of who they'll take on to fund. Solid data, proof of concept data, which I've showed you, and the ability to drive the projects forwards with the techniques and the models that we already had developed. And also clinical exposure is really important to them. So I'm a clinician, we've got access, as I say, to patient registries, international and national registries, and that makes it a lot easier to start to move these forwards, to move that clinical development pathway forward. And then, of course, appropriate patents. These people are money people, and they need to know that some of that property is protected and they won't be pipped to the post. So Purespring was set up. What was really nice was that Syncona had they helped us with set it up and actually were very keen to get really good people in right from the beginning. So Richard Francis was former CEO of Sandoz, a sort of $10 billion company. Came back to Europe to take this on. Julian has worked for over 20 years in gene therapy companies, massively well known in the field. And Ronnie has worked for about the same amount of time in renal clinical development for big companies like Astellas. So these are really solid people who are joining the team. And now it's sort of, the company looks at this with a much more commercial eye. So, what's the innovation gap and where can we fit in? And in kidney health, there's been, you can't see it very well, but just, there's been very few innovations in kidney disease in the last decades, really. The green line is kidney disease at the bottom, and the other lines are other specialities. So new therapies, new randomized controlled trials for new therapies in kidney disease really are lacking. So there's a massive unmet need here. So Purespring is taking this on, if you like, with the podocyte at the heart of its strategy. And one of the ways, so the next stage that Purespring is looking at is, how do we raise the next tranche of money? We've got the initial 45 million, that'll take us 2 or 3 years into the journey, but we need the next tranche, and how do we sell that to the financiers? And things I've learned that it takes, the amount of money it takes to get to marketing a first treatment of a gene therapy will probably be about 600 million to £1 billion. So that's the amount of money, and we want to do that in relatively few years. So that's the amount of money that needs to be raised in the next few years. So key things that we can sell to backers, but also some other followers, that the capsid, the engineered capsid that we use is unique. So that is the way that you get into the podocyte specifically. Is one of the ways is via the shell of the AAV, which is unique. Promoters, I've mentioned these molecular promoters that are cell specific, and local delivery is really important. So can you deliver directly to the kidney rather than giving it an IV injection that goes around the whole of the body? And that's important for several different reasons, including toxicity. You don't have to give such big doses, but cost of goods. These therapies are really expensive, so if you have to have a very big dose per patient, that really makes a huge difference as to the commercializability of any product. And developing the production process. This is again something I've learned is absolutely key. If you're going to give a product to a patient, it's got to be reliable, it's got to be pure, it's got to be it's got to have all the kind of quality controls that we as academics don't necessarily think about. So, and that costs a lot of money and takes time, and that's got to be done really properly. Just a quick word on the targeted delivery. So our targeted delivery is to go directly into the kidney via the renal artery, and that's not a trivial task. So that's a big part of one of the programs that we're doing in large animal experiments, etc., to get that right, so that we can get the dose down in particular, and stop these kind of off-target side effects that you might get. Other things that we're doing is licensing in other technologies. So FunCell, for example, is a technology that allows you to use libraries of AAV products. So this is a library of over 2,000 genes that are expressed by the virus that you can use to screen in a mouse model of disease to see which of those 2,000 proteins might cure the disease. In a very sort of simplistic explanation, but it's very powerful, and we can we've licensed in this technology from an academic at King's for the kind of next stage discovery, if you like, that you can then take down that pipeline to patients if you find the right therapy, the right target. So that means that we'll have several programs in development at one go, and we've already started with 3 programs at the moment in development for 3 different kidney diseases, and then the pipeline programs behind it, including the ones that are powered by those technologies that I just mentioned. So the vision is that you keep going. You don't just have one disease and you stop and you make your profit. You actually keep going, and you can, if it's successful, you'll have sort of one product coming online, and then 6 months or a year the next one, and so on and so forth, to try and keep the process moving forwards one after another. So that's the kind of landscape at the moment. We are kind of unique because we're the only gene therapy company. There are other companies working on things like small molecules and biologics like monoclonal antibodies, but gene therapy at the moment is not, we're the only company that has that area. And if you look at the gene therapy landscape, it's a buoyant environment, even in the sort of current age of slightly difficult sort of money raising in the biotech sector. So there's lots of really good data in clinical trials in some of the other diseases that I've mentioned. There's certain products already approved, and these companies are being brought up by pharma, so they're of great value in terms of the money that's available to develop them further. So I think that sort of more or less summarizes where we are. We are the first kidney-directed gene therapy company, unique focus on the podocyte, a broad pipeline with starting with 3 programs, so it's really ambitious for a biotech. And state-of-the-art therapies like state-of-the-art technologies like FunCell, and a high-quality team with Richard Francis as our CEO. And the last is Richard's slide, he sort of begin with the end in mind. So what he wants us to do is start as we're a very small biotech, but we're growing very fast as we acquire staff. But what he wants us to do is look at where we want to end up and build the team from the start that will end up with a highly successful biotech. So that's what we're hoping at very early days, and you know, I hope you can wish us well. Thank you.
We've got time for one or two questions. Are there any questions for Moin? Yes.
You said that you found there was quite a difference between the UK and the US VCs. I was just wondering if you were willing to elaborate a little bit on that.
Yeah. So the question was the difference between UK and US VCs. Clearly, my experience is relatively limited, but we were approached by at least one very big US VC, and that were almost sort of quite aggressive. They really wanted to, you know, buy the technology, etc. But I thought their approach was a bit more: we'll buy the technology, we'll buy all the patents and everything that you've got, and then we'll go off and we'll set it all up ourselves, and without really involving the, you know, that you're a bit external as the founder and the academic, and clearly I don't like that. If we want to progress in the way that I've described, where we keep on developing new innovations, then I'd like the approach of Syncona, and they've done it with their sort of the retinal companies in particular, is that they've stayed very close to the founders and the academics to help it develop. So that was the main thing that I was a little bit concerned about, but other people have said similar things about how aggressive and different cultures that the American companies might have.
Any other questions? Michelle.
Well, you've attracted some really big names, Richard Francis et al., into the company. Were these people you already knew through your research, perhaps? And if not, how did you get their attention, as at that point probably a pre-startup? How did you recruit such big names? Yeah,
so Richard wasn't somebody I knew, but Syncona sort of knew of him. They knew he was around, so they've got good networking. Ronnie, who's the chief medical officer, I had known from before, but again, Syncona did the identifying. I'm pretty well known in the field, so when people look at it and they see that I'm heading it, it gives it a degree of credibility. I don't want to big myself up, but it does help, because that, they see that it's somebody who could, it's a company that will have enough intellectual ability to go forwards, as well as the kind of, all the practical things that I've talked about. So it's a combination of things, but I think, again, it was an advantage of working with Syncona as a company, that they had some really good, a good little black book of addresses.
Good investor is a lot more than just the money.
Yeah,
yeah, absolutely.
Absolutely. Moin, thank you very much indeed.